Ready.
Bueno, muy buenas tardes. Estamos de nuevo en el ciclo de conferencias de la licenciatura en Ecología de la Escuela Nacional de Estudios Superiores, Unidad Morelia, en Morelia de la Universidad Nacional Autónoma de México. Muchísimas gracias a toda la audiencia por estar acá presente. Hoy tenemos el gran placer de presentar a ustedes al doctor Hervé Soquet del Royal Botanic Gardens and Domain Trust de Sydney, Australia, y de la Universidad de New South Wales en Sydney, Australia. El doctor Soquet es un investigador en la evolución de angiospermas y le agradecemos muchísimo su presencia y el aceptar nuestra invitación. Y hoy nos va a hablar acerca de los retos y preguntas para la reconstrucción de flores ancestrales de angiospermas. Realmente nos da muchísimo gusto que esté aquí presente. Thank you, Hervé, for being here. We really would like to thank you for accepting our invitation. And now Silvana will introduce you. Thank you very much.
Thank you.
Buenas tardes. Buenas tardes, jóvenes, estudiantes, colegas, tanto de nuestra audiencia de la licenciatura en Ecología como de otras entidades de la UNAM, otras instituciones de México, y también queremos dar la bienvenida a nuestra audiencia internacional. Posiblemente tengamos también alguna audiencia de Australia, so good morning to those Australians who are listening to us at this time. Bueno, el doctor Soquet, como bien lo dijo Mauricio, trabaja en los jardines botánicos de Sydney, Australia, donde actualmente es investigador consolidado de estos jardines botánicos. Realizó sus estudios de doctorado en la Universidad Pierre et Marie Curie en Francia, en París, que actualmente es parte de la Sorbona París 6, si bien entiendo. También realizó un postdoctorado en el Jardín Botánico de Estocolmo, en Suecia. Y bueno, es botánico, es biólogo evolutivo, y como dijo Mauricio, sus principales líneas de investigación se centran en diversos aspectos de la macroevolución de las angiospermas y de la evolución floral en diferentes sistemas. Para esto utiliza diferentes enfoques en la sistemática filogenética y comparativa, secuenciación de nueva generación, biología del desarrollo, morfología de flores, de polen y paleobiología también. Tiene más de 50 publicaciones en revistas arbitradas, capítulos de libro, así es que es un investigador de gran calibre, que además no es ajeno a México, tiene diversas colaboraciones con investigadores mexicanos, una de las más fuertes con la doctora Susana Magallón del Instituto de Biología, y algunos otros investigadores, y a partir de estas colaboraciones, si ustedes revisan el currículum y las publicaciones del doctor Soquet, verán que hay pues grandes contribuciones, ¿no?, al campo de la evolución de las angiospermas en revistas importantes de plantas y de evolución. Estamos muy agradecidos con Hervé por haber decidido participar en nuestro seminario, y particularmente a las tempranas horas de la mañana en Australia, en Sydney actualmente son las 8 de la mañana, entonces muchas gracias. Y bueno, también, bueno, gracias a toda la audiencia, y pues ya los dejo para que disfrutemos de la plática del doctor Soto. Adelante.
Thank you so much for the lovely introductions. Thank you so much also for the invitation. I'm really pleased to be giving this talk to all of you today. I should mention before I start that I told the organizers that it would be wonderful if you wanted to interrupt me during the talk. It's not a very long talk, so I will take my time, but if you have any questions, feel free to post them in the chat. And the organizers might ask these questions to me halfway or in the beginning of the talk. That might make it more interactive for all of us. So please don't hesitate. Don't hesitate to ask these questions in Spanish or Portuguese as well. We can then, I can read those languages, so it should be fine. All right, so the talk that I'm giving today is a rather broad talk on a particular topic that I'm really interested in, is the floral evolution in angiosperms. And it's based on work that I will refer to many times that was published 3 years ago, but it's just sort of thinking 3 years later, how do we think about the results we published and how did we get there? So it will be a bit more of a, let's say, a mature perspective on work that we published a while back. And it's work done in collaboration with many co-authors, and I'll mention many of them. And there are several that are actually in Mexico, as Silvana mentioned. All right, so let's start. So what I'm really passionate about is the extraordinary diversity of flowers in flowering plants. I should mention this picture is actually assembled by our colleague Santiago Ramirez Barahona for recent work that we did together. And if we posted a picture like this about leaves or roots, it wouldn't be as spectacular. So we're all fascinated by that. For many centuries, that floral diversity has been the key to identifying plants. And basically, the general idea that's never really been fully tested is that it probably has something to do with the extraordinary diversity of species, the incredible species richness of flowering plants themselves. And so all of my work is trying to understand what is the link between this extraordinary story of diversification of floral shapes in flowering plants since their origin and the diversity of flowering plants that we see today. I don't think we'll ever answer that question entirely, but we can try, and this is what I'm trying to do through my work. In the first part of my talk, I will mostly focus on one particular question, which is probably also the question that is most difficult to answer. And it's looking back at the very first flower, or let's say the flower of the most recent common ancestor of all flowering plants. What might have it look like? And this is an important question because it will tell us something about where flowers came from within land plants, and also why flowering plants suddenly appeared and what was so important for them to diversify so quickly. So I should specify that this question has been asked for a very long time. In fact, back in 1907, more than 100 years ago, there were two competing theories. One that is summarized by the first row of pictures here is that it was a rather complex flower with many parts, such as we see today in the magnolia, many tepals, many stamens, many carpels, but we see also in other flowering plants. And this was argued on various grounds, but in particular, the fossil record was important in recognizing that these may have been close to what the ancestral flower looked like. I should mention at this stage, we would never, ever again look among living members of a clade for identical pictures of what they look like 200 million years ago, 150 million years ago. But these comparisons have been made over and over. So it's important to take from a historical point of view that magnolias and their relatives have been proposed as primitive by many people in previous times. And these were also superficially looking like an important group in the fossil record called the Benetitales. And you see a reconstruction here. These are the flowers. And they do have bisexual reproductive structures that in many respects look like modern flowers. And so this comparison was made. However, nowadays, no one thinks of Benetitales as actually having flowers. People think of them more as having independently evolved reproductive structures that do look like flowers. But at the same time, there was a competing theory that instead, the early flowers were very simple and perhaps unisexual. And I've put a few examples here. The one on the left-hand side is a picture that I took in my former university in Université Paris Sud, in Orsay, in the suburbs of Paris. And there is a lot of temperate trees around there. And most of the temperate trees that we have in Europe have very simple flowers, such as in the species here of beech. At the same time, there was also even more extreme examples of simple flowers, such as the flowers of Chloranthaceae. So in Chloranthaceae, it's a very unique family. In 3 of the 4 genera, the flowers are as simple as you can get. There's absolutely no perianth, and the flower is either one carpel or 1 or perhaps 1.5 stamen. So you can't be more simple as that for a flower. And this was, the Chloranthaceae were put back to the forefront of these theories when their fossil pollen was found as one of the earliest fossil pollen ever found for flowering plants. At the same time, in about 20 years ago, fossils were also found. Some of the earliest fossils of flowering plants were found as aquatic fossils with very simple inflorescences, such as Archifractus. And this also put back on the table the idea that, well, in fact, the earliest flowers may have been very simple. So at the end, we actually didn't know, and we'll see what we now know. But these two theories have competed for a really long time. However, nowadays, we are in a completely different setting. We have a really thorough understanding of phylogenetic relationships in flowering plants. So this is a very simplified tree that shows the main lineages of flowering plants. And as you might know, most of the species diversity is in these 2 major clades, eudicots and monocots. That's about 95% of all of the species of flowering plants. But since 1999, we know with quite good confidence, and this has been confirmed many, many times, that the earliest diverging lineages of flowering plants are first the species Amborella trichopoda from New Caledonia, then the order Nymphaeales, the water lilies, and then a very strange order called Astrobelliales. And I've put you here an example. It's actually a very strange species. So Astrobelliales includes, I'm not exactly sure, but about 100 species. But many of them are in the southern hemisphere, and in particular, it's named after Astrobellia scandens. And this is a vine from the northern tropics in Australia. It's a very weird flower. It stinks. It attracts carrion flies for pollination, and you really don't want to smell that flower. So if one of you ever comes to the Botanic Gardens in Sydney, let me know and I'll show you, because that blooms in our garden. And why am I showing you this picture? Because although we have a very thorough understanding of the phylogeny of flowering plants, this is not enough to understand what the earliest flowers look like. We would never anymore think that because Amborella is the sister group of all remaining flowering plants, that means that this is a portrait of the ancestral flowering plants. This is not a good way to read a phylogeny. In fact, there's a lot of time. There's an extreme amount of time from the origin of flowering plants to Amborella for evolution to have occurred. So we should be very careful in drawing any conclusions about what Amborella is compared to the origin of flowering plants. And to show you a few characters, the flowers are entirely spiral, and more importantly, they're unisexual. So we have female flowers on the left-hand side and male flowers here, and they're just, the sexes are separate. So that didn't help in solving the question about whether the ancestral flower was bisexual or unisexual. Because of that situation, it made reconstructions very uncertain at the base of angiosperms. So I mentioned that the arrangement of parts was spiral, but in the next lineage, in Nymphaeolus, it's actually whorled. Many people think of water lilies as spiral, but it's not true. And you see it really well in that picture. You see various whorls of 4 parts. The next one, however, Astrobeliales, is spiral again. So that makes a very complicated mix, a combination of characters at the base of angiosperms that has made reconstructions very ambiguous for a long time. And I'll show you one example here. It's truly groundbreaking work by 2 of our colleagues. So on the left-hand side, Jim Doyle, who works at the University of California in Davis, and on the right-hand side, Peter Andres from the Institute of Systematic Botany in Zurich at the University of Zurich. And together, for a very long time, they have put together morphological matrices of so-called basal angiosperms. So all of the early diverging lineages of angiosperms, plus all the magnolids and some early diverging lineages of monocots and eudicots. And they used these new phylogenies obtained from DNA sequence data to reconstruct the evolution of key characters, including floral characters across the flowering plants. So this is one example from one of their publications. They use only parsimony in this data set. And what they found is possibly that the ancestor of all flowering plants had many parts, as you can see here in these floral diagrams. I'm sorry, I'm struggling a little bit with my cursor, but do you see my cursor? Yeah. So at the base here, you see 2 possible floral diagrams reconstructing many tepals, stamens, and a few carpels. But you see the arrangement was completely uncertain, was ambiguous. There was one spiral version on the left-hand side and one whorled version on the right-hand side. So when we started this work about 10 years ago, we had a lot of admiration for that work, but we thought we could take it an extra step further. We wanted to sample more flowering plants. We wanted to score traits for species, not for higher taxa. As you can see here on the tips of the tree, we have some genera, but also entire families. I'll show you an example here. Lauraceae, arguably Lauraceae are quite constant in some of their characters, but that's 3,000 species. And so here Lauraceae are scored with their presumed ancestral states. And so that sort of assumes that we know what the ancestral states of Lauraceae might have been. And in fact, we don't really know in many cases. So we wanted to skip that level of assumption and go through a direct inference by scoring only species. And we also wanted to apply some alternative methods for reconstructing traits. So before I continue, I should specify more clearly what we're talking about here. So when I talk about the ancestral flower in this talk, I'll be referring to the crown node of angiosperms. And so in phylogenetic analysis, the crown node is the most recent common ancestor of all living members of flowering plants. It is not the only ancestor. In fact, there are ancestors all the way down the stem lineage. There is a possible infinity of ancestors. We don't know what they look like. We don't know how many species we may have counted here, but it's just to show there's many ancestors. So there's the crown node and then all the way back to the stem node, which is the divergence with their sister group. There's a lot of uncertainty here. So I'll just be focusing on that crown node. That figure is taken from a review that Susana Magayan and I wrote a few years ago to remind ourselves that there's so many key questions in angiosperm macroevolution that remain entirely unsolved. And some of these questions illustrated on that figure are, in particular, the age of the crown node of angiosperms. In my opinion, not everyone would agree with that, is entirely unknown at this stage. It's somewhere between 140 and 250 million years. That we can probably say, but many people disagree on the actual age. Some people think it's very old, other people think it's very young. In fact, I think we still don't know. But the other important thing that we don't know is how the different characters that define the flowering plants, the bisexual flowers with the perianth, the double fertilization, the vessels, there's many characters. How did these assemble along the stem lineage? In which order they came? That remains a completely unknown question. And the reason for that is we don't have any stem lineage fossils along that line. We have a few groups of fossil seed plants that might be related to angiosperms. I mentioned Bennettitalis earlier, glossopterids are another one. But we don't really have angiosperm looking like fossils along the stem lineage. So a lot of questions remain, but luckily we have good understanding of the phylogeny and we can answer already some questions. And this is what I'm trying to do here. So the paper I wanted to talk to you about was this paper that we published in 2017 called The Ancestral Flower of Angiosperms and Its Early Diversification. And one of the figures of this paper is the one here on the right-hand side. It is a 3D model that was based on our results from our reconstruction. So some of you might have seen it, maybe not. It became quite controversial, and I'll go back to this later in my talk. But what I want to tell you here is the story of how we came to reconstruct that model and what our methods were and what the questions remaining are. I just want to ask the organizers, is there any question yet that I might answer at this stage?
No, I don't see any questions at the moment in the chat.
No worries. Okay.
Les recuerdo a nuestra audiencia que pueden hacer preguntas durante la plática y las pueden poner en el chat y podemos hacer estas pausas de cuando en cuando para responder preguntas particulares. Gracias.
Thank you. So please do not hesitate. I'll be more than happy to stop. So to get to this point, actually, so 10 years ago, when I was still working in Paris, I had these dreams that all those things I wanted to do, and I sort of took a really strong risk at that time, was to undertake a project that will never finish, and I call it the E-Flower project, and it's a career-long objective. So it will continue as long as I'm alive, probably. And it started on a grant proposal that we didn't get. So I assembled together 14 people that I knew who were experts in various aspects of angiosperm phylogeny and floral morphology. We didn't get the grant, but we decided, well, let's do it anyway with some of these people. And this is where we are now. So the whole idea was that this was back in 2010, 2011, we had already a remarkable understanding of relationships among flowering plants. This was a simplified tree that I did at that time. I have never redone that figure since then, but already back then, molecular data had helped solve most relationships among families of flowering plants. This is a tree where every tip is a family. There's about 400 families of flowering plants. Of course, there's a lot of uncertainties remaining in particular details, especially in shallow nodes, some deeper relationships as well. But it is very precious knowledge that we didn't have 20 or 30 years ago. And it is really thanks to the molecular revolution in phylogenetics. At the same time, for a very long time, for more than 200 years, flowers have been looked at, observed, described in taxonomic descriptions, and more importantly, in the last 50 years, considerably studied with new tools such as scanning electron microscopy and currently now X-ray tomography. So there's an enormous amount of knowledge on floral structure, and so the whole basis of the whole point of E-Flower is to put these two together: what we know about flowers and what we know about phylogeny together, to reconstruct a better understanding of floral evolution through deep time. I should mention that a lot of people in the world are studying floral evolution from many different angles. This is not the only way to study floral evolution. Obviously, studying plant pollinator networks is really important knowledge. But what I should mention is perhaps most of the knowledge that we do have about floral evolution at the moment is mostly in very focused clades of the phylogeny. So particular genera, particular families, there's a lot of studies there, a lot of information has been given, but the deeper you go in time, the more questions remain. And so this is what eFlower wanted to address. So we wanted to address quite a few questions, but some of these that I've written down here is what were flowers like, not just at the root node of angiosperms, but also in different clades of angiosperms. How are floral traits correlated with one another, and also how traits influence diversification rates. So for that first paper, we assembled a data set that was with a sampling strategy that was based on the groundbreaking work that Susanna published with other colleagues in 2015 in New Phytologies, which was at that time the best data tree, in my opinion, of flowering plants that was available out there. We actually started our work, our collaboration 2 years before. So 2 years before she published that tree, we agreed that we would score the same floral traits for every one of the species in that tree. The reason why that tree was particularly good compared to the collection of trees available at that time was that it had a very good sampling of flowering plants, 792 species, which is a lot, but at the same time, small enough that it was a goal that we could tackle. And it represented most of the families recognized at that time and most of the orders. And more importantly, the time component of this tree was particularly well done because it was based on more than 100 fossil calibrations. So what we did is we collected information on 27 floral traits that we recorded in a database that I developed for the purpose of that project called Proteus. And a key aspect of this database is it's a multi-author database. So many people collaborate on different projects. This particular data set was built actually by 47 different people, some of them contributing more than others. And importantly, every single record is tracked to an explicit source. So if you ask me, where did you get that information on that species? I think that's wrong. I can tell you in less than a minute exactly who did it, when, and from what source. And that's been really, really useful in us improving the quality of this data set over time. So this particular data set was based on 13,000 observations linked to almost 1,000 explicit references. And I should mention, actually, the oldest reference was from Jussieu, I think, a French botanist who was already reporting information on flowers at the time of the French Revolution. And it's just to show you how information in systematics has a really long life cycle. We still are using information from very, very old descriptions. So this is just a snapshot of the principle of Proteus, the database that we used to build this data set. And on the top level is a traditional morphological matrix with a sort of a 2x2 table of taxa and characters. But what Proteus is adding is this depth of observation. So instead of just having one single score, we're recording all of the data that are contributing to the summary of a character value for a given taxon. And so what we're tracking again that's specific to Proteus is references and users and when they're recording the data. And lastly, just to show you, it's been growing quite slowly but steadily over time. More than 100 people have used it so far. It's still very much alive at the moment for various projects. Now I want to tell you a little bit about this little bump in history, this 2013 boost in data. How did that happen? So the story is that we were starting to do that project. We had these already quite ambitious goals of scoring every family of flowering plants. And we realized soon enough that we weren't enough people. It was a lot of time. Every time we look for data, it can take hours just to find the correct information on a particular species. So we decided to call for help. And what we did was to invent a new system that we call the eFlower Summer School. And the whole idea was that we invited about 15 students from all over the world, mostly Europe, but one person came from China, for instance, and another one from Brazil. and another one from Mexico. The whole idea was that we would invite them to come and work for us, input hardcore data of floral traits in our database for 6 days, locked in this beautiful room at the University of Vienna. But in exchange, we paid their travels. We paid their food, their drinks. We had seminars every day to stop the rhythm of data scoring. So these were invited speakers. Susanna came over for that particular first summer school as an invited speaker, but also other colleagues. And in exchange, we also promised them co-authorship of the paper. So it was a good deal for everyone, and it still to this day was one of the most fantastic experiences of my professional life, to have this live experiment with all of those wonderful people, many of whom have gone on to take academic careers now, and some of them are collaborating together without us. So it's been really nice interactions with all those people. What we did with these, so we ended up entering actually only 50% of the data in this 1 week, and the other 50% took another 2 years of completion, curation, etc. But it really helped us get a start. A little word about the methods that we used. We used 3 methods for reconstructing ancestral states, so parsimony, but also maximum likelihood, and a Bayesian approach called reversible jump Markov chain Monte Carlo. I'll explain very briefly what this means in a few minutes. All of these methods take data so a data matrix in a phylogeny to make an inference about reconstructed ancestral states. None of them is perfect, but together they might give us a sense of confidence in the results. We also use different backbones for the molecular phylogeny of flowering plants. So the main one was a tree from Magayan and all, but we also altered this tree in various ways to take into account some additional relationships that are still uncertain, in particular, the relationship between monocots, eudicots, and magnolids. So in the end, we didn't do 1 analysis, we did 5, and in fact, we did 10 because we repeated every analysis with either angiosperms being young or very old, just to test the sensitivity of our results to these particular assumptions. I'll start with one key result that I think is paradoxically perhaps our strongest result. It is a result that is not surprising, but that yet was absolutely uncertain until we published that study. And it is the sex of the ancestral flower. So our results very strongly support that the first flowering plants, or at least the most recent common ancestor of all flowering plants, had bisexual flowers, such as this little diagram here with both a gynoecium and an andrecium, so carpels and stamens in the same reproductive structure. In that many times throughout the history of flowering plants, flowers have become secondarily, have become unisexual, and sometimes they have also reversed back to bisexuality. And so this is the full tree that you see. The colors are indicating the particular character states. And as you can see from the pie charts here, we get like very strong confidence in that result from the model-based approaches. I should mention that because Amberella, which you can't see here, but it's hidden, it's like the very first line you see, it's blue. Because Amberella is unisexual, that means that parsimony and reconstructions are still ambiguous, as they have for forever. So if we rely on parsimony to reconstruct this result, we still don't know. If we take a model-based approach to this problem, we have an answer. And so what is the right answer? We probably don't know, but in that case, I should explain why this is different. So these model-based approaches, one of the key advantages, they have a lot of limitations. So they are based on models that have limitations, assumptions, etc. But their main value is that they do take time into account. So this result is based on the method allowing itself at least 140 million years for the sex to change. And we know sex have changed many times throughout the evolution of flowering plants. So why could it not have changed in Amberella? In that case, everyone is absolutely ready to accept that Amborella has changed its sex over this time for various reasons. So first of all, the female flowers of Amborella actually have remnants of male parts. They have staminodes, sterile stamens. And secondly, there have been observations in greenhouses, in particular, I think in a greenhouse in Lyon, by colleagues, the group of Charlie Scott, who have observed that there are actually bisexual flowers that can be produced. So they have demonstrated that sex in flowers of Amborella is actually fluid, and in some conditions, they can be bisexual flowers. So it really shows that Amborella has still the capacity of producing bisexual flowers, and it is very, very likely, this is probably the result that I would be very surprised if it was overturned in the future, that the ancestral flower was bisexual. Here's another example that we consider very strong from our study. It's the reconstructed ancestor from a very large clade of flowering plants, which colleagues in 2007 have proposed a new name for, the pentapetalae. So it's highlighted here in these yellow to red colors. You see it's about 75% of the diversity of flowering plants. It is more or less what was called for a long time the core eudicots. So a very core part of the eudicot clade. And most of these flowers have either 5, sometimes 4 or 3 parts in their perianth and stamens, and they're also whorled, and they also have a differentiated perianth with sepals and petals, which I've represented in 2 colors here. So what we have here on the right-hand side is just one of our characters, and that character is the merism, so how many parts per whorl for the perianth, the sterile parts of the flower, so the petals and sepals. And we found that the perianth, the ancestral perianth of pentapetalae, was pentamerous. And we found that in every single analysis that we did, whether it was parsimony, MP, maximum likelihood, or this Bayesian approach that we did. And when we could get some scores of confidence in likelihood and Bayesian approaches, these scores were very high. These are the the proportional marginal likelihoods post-reconstruction. You see they're very close to 1, so it's very strong support. But more importantly, in the Bayesian approach, over 10 million generations, these probabilities are centered around a very, very high probability. So these are 95% credibility intervals. And these take into account phylogenetic uncertainty, so different relationships, different times, different values for the parameters of the models, and also even the model is varying through these analyses. Just before I explain that a little further, we have other reconstructed characters for this clade. So we reconstructed also as having 2 whorls of stamens, 5 and 5, and 5 fused carpels. So we've decided to represent some of the results with these botanical diagrams and also this little floral formula here. It is not a surprising result at all. Many people would have predicted it, but it's kind of nice and cute to finally have analytical confirmation of something that people have presented for a long time by even calling that clade pentapetalae. It was never really demonstrated that pentapetalae were actually ancestrally pentamerous. So we were quite happy to find that result. So just to explain a little bit what the reversible jump MCMC is doing, in that particular case, it was not possible to just have a binary character. There's too many conditions. So there are dimerous, trimerous, tetramerous, and pentamerous perianth in flowering plants. There's more, but these are the main states. So the simplest model had 4 different states, and that makes 12 different parameters in the most free model. So this is an example here. This is our Q matrix that's linked with this model and has 12 different rate parameters. That's a lot of information to estimate from the data. And so traditional likelihood approaches, what they do is they fix the model with more or less parameters. So they can either assume that all are equal or they're all different. These are the 2 extremes. And we conduct an analysis and we get a result. What's not satisfying here is there's a lot of models in between. In fact, there's 27 million models. that you can create by making two parameters equal or forbidding some transition, putting them to 0. So what the Bayesian approach that we use in this paper does is quite remarkable. We didn't design that. That was implemented in a program called Bayes traits, which was designed by Mark Pedro and colleagues quite a while back. But we applied it to this data set. And what it does is that it integrates over many models. So it takes off the load of having to assume what the model is, and it integrates over these many models. So just skipping down a few notes, going back to the ancestral flowering plants, this is what we obtained if we just looked at the most likely results, the best states that we obtain across different analyses. So we reconstructed the most recent common ancestor of all flowering plants as having a world perianth, not a spiral perianth, with many parts, more than 10, and these parts are arranged in worlds of 3. So if you take more than 10 world and arrange in worlds of 3, that's one of the possibilities, to have 4 concentric worlds, as I've drawn here for the perianth. There could have been more. And we reconstruct actually the same structure for the andricium, at least 4 worlds of 3 parts each for the stamens. And surprisingly, we found the carpels to actually be spiral in these first analysis. So what we did here, we weren't quite sure about the significance of these results. And we'll never know if they're true. We'll never be sure. But what we decided is to take our results as they were, without applying our filters of what we thought it should be, and publish that. I should mention at this stage, there's a lot of uncertainty remaining to some of these particular traits. In particular, the phyllotaxis, the arrangement of parts of spiral world, remain quite uncertain. We knew that. But instead of choosing spiral over world, because that's what people thought it was, we decided to publish what our results were saying, which according to many morphologists actually is not absurd at all. And so that's how we came to these conclusions. And we decided to communicate with the greater public by making a 3D reconstruction of that. And I'll explain it in a few minutes how that was done. So just to summarize our study, we were able to infer ancestral states for different keynotes. We focused on 15 key named clades of the flowering plant phylogeny. And this is another figure from that paper where we have these reconstructed floral diagrams for the Angiosperms, but also the pentapetalae. I mentioned that a little earlier. We have a very interesting hypothesis for the eudicots that might have started as dimerous, which you do see in some early diverging eudicots, such as the Papaveraceae, for instance. And then we have the monocots with their more simplified trimerous model that's very easily derived from the ancestor of flowering plants. So this has allowed us to rethink floral evolution I think, quite a different way. Because if this is true, we still don't know if it's true. But if these were world flowers at the very start of flowering plants, it's easy to shed some whorls, to reduce the flowers to get to the monocots. To get to the magnolids, we reconstruct the same ancestor, but within magnolids, we also shed some whorls to get flowers of magnolias, Annonaceae, Lauraceae. These are really easily derived from these ancestors. To get to the pentapetalae, however, it gets a bit more complicated. Our results are suggesting that perhaps instead of shedding, you are reducing the merism from 3 to 2, and then something happens from this model to the model of pentapetalae. And one of the things that possibly may have happened is a fusion of the whorls. So it is not impossible that the corolla of the pentapetalae is the result of fusion of the 2 inner whorls of the ancestral eudicots and possibly the 2 inner whorls of angiosperms. And if that's the case, it's also not impossible that the perianth remaining in monocots is just the original two inner whorls. There's other possibilities. It could be the two outer whorls or something else. But it would mean that the corolla of the pentapedally perhaps is not homologous to the corolla or the inner whorl of monocots, but perhaps it's homologous to the entire perianth of monocots, which would not surprise Evo-Devo specialists, which for a long time have found that B-class expression genes are their domains of expression is very similar across the entire perianth of monocots to that found in the corolla of eudicots. So I'm not sure I'm doing good justice to what I'm saying here, but I just want to explain that this study has allowed us to challenge quite a few established dogmas on floral evolution. And a lot of the comparative studies that are being done, a lot of the interpretation of Evo-Devo studies are sometimes made on assumptions of homology between flowers that in fact have never really been established, at least not based on ancestral state reconstructions. So the power of that study, in my opinion, is not to have delivered final results, but to have allowed us to rethink floral evolution in possible alternative ways that might help us progress. I hope that doesn't sound too arrogant, but I just wanted to express where I think we have arrived at this stage. And just to finish on that part, I wanted to mention that, of course, the fossil record should, in theory, hold some key clues about the origin of flowering plants. And some of my colleagues might even think these are the only clues. We shouldn't even bother looking at the phylogeny. The problem is that the oldest fossils of flowering plants are no older than 130 million years old. That is a fact that not everyone agrees with. Every now and then there are reports of a Jurassic flower, but these are very quickly dismissed by a majority of the paleobotanical community. So at this stage, we really don't know for sure that there has been any fossils of flowering plants, at least of crown flowering plants, before 130 million years. There might be some stem lineage pollen grains, but no flowers. So because the ancestor of all flowering plants is at least 140 million years, you cannot use the fossil record to have a direct confirmation of the ancestral flower. Maybe we'll find that fossil and maybe it will completely invalidate my results, or maybe we'll suggest that some of it was correct. But what we did in that paper was to have just a quick look at what we knew of the fossil flowers once they arrived. They arrived in the second part of the early Cretaceous. And what's remarkable about the earliest fossil record that we know in terms of flowers is that it was already very diverse. As you can see here, there's very different structures. This picture is taken from a groundbreaking review by Pat Herendeen and all in 2017. But it's largely based also on the collaborative work by Elsa Marie-Fries, Peter Crane, and Kai Pedersen that they published in a book in 2011. And you see these flowers here. They're diverse, but there's a majority of actually whorled flowers, whorled trimerous flowers. There's also the odd pentamerous one, and there's one or two spiral flowers, also very simple flowers. So we think this does not confirm our results, but it is compatible with the prediction that we would have from the scenario, which would say that by 130 million years, we already expect quite a few diverse floral morphologies, including possibly a majority of whorled flowers. So there's big questions remaining here. We haven't put together the fossils with our reconstructions. The fossils have been only used as calibration points for dating so far. And so it will be really interesting to see when we have the fossils in the phylogenies, what the results, how the results are going to be affected. I'll just take a mini pause here. Are there any questions so far in the chat?
No, we don't have any questions in the chat. We only have greetings from Honduras. And we have about 70 people in the audience.
Okay, wonderful. Yeah, so as expected, I'm I'm taking my time, and it's taking longer than expected. So it was good that I planned on 30 minutes because it's already been 45 minutes. So what I'd like to do in the second part is just take a step back and think a bit about the science communication aspect of that story. So as I mentioned to you, we thought that floral diagrams wouldn't speak much to the public. And so we decided to take the risk of creating this 3D model. And I mentioned that it was a risk because for several reasons. First of all, there was a lot of uncertainty in our results. I've highlighted here a few of them. So I've said that the bisexual flower was highly confident result, but when you see the information on other characters, such as the fact that it was whorl, it's less certain. So when you do one model, instead of the actual infinity of possibilities that could be inferred from our results, we take the risk of people having the false idea that we know for sure what the flower was like. But as anything in science, it's a hypothesis. It's a working hypothesis that helps us think and visualize what the results might have meant. The second risk that we took is that by drawing a 3D model, we had to take arbitrary choices on things that we have not reconstructed. This includes the shapes, the relative sizes of the organs, and the colors. So we have no idea what color it was. These are absolutely arbitrary colors, and so are the shapes. The funny thing, though, is that it really worked. It really worked in helping people relate to this result. And we wrote quite a few, we wrote a media release, but we also translated it into 6 languages because we had a lot of co-authors. We had 36 co-authors on the paper, including all the students from the summer school. So we wanted to reach out a broad community. And the media really liked it for some reason. They picked up on it. It was a strange moment for me because I had just moved to Sydney. I hadn't started my new job in Sydney. And I was there by myself at the other end of the world, having to sell the results from the work that I did in the last 10 years. It was also, sadly, the paper came out after the last International Botanical Congress in Shenzhen. So many of us were there, and we were hoping that the news would be released during the Congress because it would have been great. But no, it was released just a week after. But the media liked it, and these are just a few examples of the titles that came. And thinking back at it, I think no one in the media or in the broader public really worried about the phyllotaxis of the perianth, of the ancestral flower. What they learned primarily is that it may have been bisexual, or more importantly, I think a lot of the people, by reading the story, realized suddenly that all flowering plants at some point had one ancestor. They all came from one spot. And so it was a really interesting exercise in science communication where we did a study for particular reasons, found a result, and that was a vector to teach something completely different to people about evolution. Of course, the fact that all flowering plants are monophyletic and came from one common ancestor is not a result of our study. It was actually an assumption that's consistent with all phylogenetic data that had been obtained so far. But it's interesting what came out in this. But clearly the image was very important in this vector. And I think on the bottom right is one of the headlines that I was most glad about. It was the LGBTQ newspaper, which decided that it was so cool that the first flower was bisexual. No one actually realized that most flowers are bisexual. And so this was another interesting effect of that study, is to make people realize how different flowering plants are and how diverse they are as well. And just to provide some statistics, there were a lot of original stories written, many radio interviews that we did in 24 languages in 38 countries. It was my most interesting experience with the media, and I learned a lot through that. But the key message here, actually, if any of you has or will be involved in such experiments to sell your paper to the media, you have to be really proactive. None of this happened by chance. we thought we had a chance to talk to the media with this particular story. So we looked for the attention, we wrote the press releases. Otherwise, it's unlikely that they would have heard about it. And it's not just it might be perceived as an exercise to make people talk about your work and sell yourself to other colleagues. And part of it might be true, but most importantly, it's also a rare opportunity to talk to the public about science, about modern science. That's why we did it primarily. And then it went just mad in the next few years. There were artists that decided to create those 3D models to 3D print the 3D models. So here's an example with in a museum in Bonn, I think in Germany. A couple of years ago, these are the two kids of two of my co-authors, Jörg and Maria, and some of their family. And these are the little ancestral flowers being reproduced as an artist exhibition. And the background is the one from my intro slide. And it's another artist who decided to draw a Cretaceous landscape. There's quite a few scientific mistakes in there. But interestingly, you see the ancestral flower here is in this little bush here. Rebecca Hernandez, who did a PhD with Susanna at UNAM, this is Susanna, had an ancestral floral cake for her dissertation. So that was really lovely to see. And one day, randomly, I saw that one person in a lab in France working on floral evolution had decided to dress as an ancestral flower with this hat for a party. And I really like the caption that I found also. There's a black box for the unknown ancestors. That's her T-shirt. I tried to put that on the back of my back 2 weeks ago at a party. The theme was primitive, and I had no idea what to dress. I decided to put a picture of the ancestral flower, but no one really cared then. So it was like the wrong crowd. So how did we get there? I just wanted to tell you the story of drawing this diagram. Initially, I really wanted to do a digital drawing. I didn't have those skills, so I asked around for illustrators what they thought. And we've had this great colleague, Agathe Evermans, who's a scientific illustrator at the French Museum of Natural History in Paris, and was the partner of one of the co-authors. And she said, I'll do it, but I'll do it my way. And her way was to first do a drawing to visualize a little bit more, and then do a life-size sculpture. You see my bag here. The sculpture was really gigantic, but it was the way that she could actually visualize and modify the parts. I had to come many times to the museum to organize the parts so they would actually be whorled and in whorls of 3. It was quite a funny exercise to play with the ancestral flower life size. And so she used this adult Play-Doh called plastiline and arranged those parts. We took a picture of it many times. We discussed it with co-authors. And of course, some co-authors thought it would look completely different, even within the same realm of results. And at that stage, we took the decision also, let's not worry about it. It is an artist's impression. There's many ways to interpret that. Let's just let her do the way she visualizes this flower. Eventually, the flower was scanned. So it wasn't CT scanned because of the material that was used to this flower, but instead it was, we used a method called photogrammetry, I think, which is used to scan some skulls by some vertebrate anatomists. And so we took thousands of pictures, and then a computer model was created based on these pictures. And this is what we obtained. And then it was colored by other colleagues in Vienna. So it was a real collaborative work by people involved in at least 3 different countries to get to that point. Again, a lot of arbitrariness was placed here, the colors, the shapes. I insisted that Agathe should never look at particular living flowers in the process, but it is not impossible that water lilies or magnolias were in her mind when she put these shapes together. So people have expressed the resemblance of our model with magnolias or water lilies, but truly there's many differences and we shouldn't make these conclusions. And just to finish on science communication, so this was a very interesting poster that was printed in a giant size in the corridor of the metro in Paris-Montparnasse, and I never saw it. This appeared a year after I moved to Sydney. But it's to show you how publicized this model has been. This was led by the CNRS in France, who had the image and then asked if they could borrow it for this long poster. So we know this model was probably wrong. There's probably a lot of results that will be refined in the future. I still think it was worth the risk because people talked about it. It generated a lot of ideas. People have written entire papers about whether or not it was spiral or whorled. We still don't know, but I believe this conversation has been very productive to the state and will probably help us refine this work in the future. Any questions, or shall I just continue?
There are some questions about the previous part of the talk. I don't know if you prefer to just finish this part and then we'll have questions at the end.
Yeah, probably. In that case, I'll probably wait until the end. Yeah.
All right.
So just to finish, I'd like to share a few perspectives on what we're up to at the moment. There's various new projects that have derived from that, from our perspective, but the key one is what we call paleo e-flower. So we're focusing on the fossil record and trying to put that in the picture, in the broader picture of floral evolution in angiosperms. And this work is done with my two key partners since the beginning. So Jörg Schönenberger and Maria von Balthasar, who work at the University of Vienna, who helped me, who co-ran the first e-flower summer school with me, and also are really complementary in expertise to mine. So they know the fossil record much better than I do, but I also know flowers much better than I do. So what I brought in this was more the expertise on doing the analysis of ancestral state reconstruction, putting the phylogeny together, but they brought the real knowledge about the flowers. And so what we want to know is where fossil flowers fit in the phylogeny of flowering plants, and also ultimately what they can tell us about floral evolution once they're in that framework. And just as an example, is a paper that we published just a few months ago in the American Journal of Botany. It was a proof of concept of a new approach to conduct phylogenetic analysis of fossil flowers. And just to summarize, the method that we use in this paper was not novel at all. It has been used by various colleagues for a long time. But what we did for the first time, more or less in that paper, was to conduct analysis where fossils could go anywhere in the phylogeny of flowering plants. So we used our e-flower data sets as support to conduct a novel type of phylogenetic analysis where we don't assume that the fossil is in a particular clade before we conduct any study or analysis. And so what we did is we used this background, as I said, the same tree, the same data sets. We expanded the data set a little bit. We added a few traits, in particular some pollen traits, and we scored 10 fossils in that paper for the same traits. So it's a combination of new fossil data with the previous data set from e-flower. And what we conducted were so-called molecular backbone or molecular scaffold analysis. So we fixed the phylogeny of flowering plants. This completely fixed tree. All of the relationships among extant species cannot change. And then we use parsimony to get a score of where the fossil fits. So we actually test every single position in the tree. There's 1,582. And I wrote a little R script to do that, and it's actually really simple. And so we allow the fossil to go anywhere in the tree and see what happens. Just to show you an example, this is a fossil flower called Moldinia, described from the mid-Cretaceous of Germany. And the flower, if you look at it, actually is quite reminiscent of living flowers of family Lauraceae, which tends to have two whorls of perianth. You might see here in this reconstruction one whorl, and there's one whorl in the back, and also two or more whorls of stamens. And importantly, the stamens here have basal appendages, which are nectaries that are typical of Lauraceae, and just a single carpel in the center. So the morphology of that fossil really highlighted the relationship with Lauraceae, but it was never really tested in any full analysis of all flowering plants. And so we scored 22 characters. These are not all of the characters that you can observe in the flower. They're just the characters that we had in our data set. This is the previous assignment. And when we ran the analysis, we found only 5 most parsimonious positions, and all of them are in or very close to Lauraceae. Here's a little detail of the actual phylogeny. So this is family Lauraceae, goes in 2 of the internal branches, also the stem branch of Lauraceae, possibly the stem branch of the sister group of Lauraceae, which is Monimiaceae and Hernandiaceae. So it's just to show like how powerful these analyses can be. Obviously, we don't know the answer, but there's a lot of good reasons to think that this is Lauraceae, including these basal nectaries, which we didn't score in our analysis. So it was very reassuring to see that if you allow this fossil, which to some of you might look like a monocots, to go anywhere in the tree, including in the monocots, in the eudicots, anywhere, it actually goes in the Lauraceae. And so even with that few characters, we have some interesting power of discovery of finding where fossil fits. But here's another example. This is the so-called Rose Creek flower that was known for a long time, but Steve Manchester and colleagues recently formally described as Daquotanthus. And it is a fully pentamerous flower with 2 whorls. Here are the petals, there's the sepals in the background, and there's 10 stamens and 5 carpels that are fused. It might remind you of something we saw earlier today. When we do the analysis, we find them in various positions in the pentapetalae. We do find them close to the fabalis, which is where the authors said it should be, but we find many other positions, 24 in total. We don't know, again, what the answer is, but the power of such an analysis is to suggest many other positions that perhaps some authors might not have thought about. And to be honest, it is absolutely impossible for anyone to have perfect knowledge of the entire diversity of flowering plants, to do these hand scans, to think of every possible family that a fossil might be related to and look at it more closely. So we think of these so-called paleoeflower phyloscans as sort of a first-round analysis to discover where a fossil might be before we look closer at some relationships. Interestingly, however, the floral diagram of Daquotanthus is really, really close to that reconstructed for pentapetalae. And so it also highlights another property of these analyses. In the past, many fossil flowers have been attempted to solve to be a member of a family or of an extant order of flowering plants. But the reality is that some of these oldest fossil flowers, they might actually be on very deep lineages, on stem lineages of very broad clades. And with this tool, we can possibly identify these relationships. So what's next? We're building a bigger data set. And for this, we're using a new data tree that we recently published with Santiago Ramirez Barahona, who works at UNAM, and also together with Susana Magayon. And what we did there is we built a better tree with all of the families, 100% of the families sampled, including the crown nodes of as many families as we could, all of the orders, and what we think is the largest number of fossil calibrations ever attempted. So I'm not giving a talk on this. I think that actually Santiago gave a talk recently in your same seminar series, but I just want to tell you what we're doing now is scoring every species in that tree in our eFlower data sets so we can improve the sampling. We also conducted a new summer school in Virginia 2 years ago to help us build this new data set, where we added a stronger teaching component. So we taught molecular dating, ancestral cell reconstruction methods, in addition to getting data in the data sets. And we were about to have a new one in Sydney a year ago, and unfortunately we had to cancel it 2 weeks before the start because of COVID. And a key actor in what we're trying to do now in the Paleoflora project is the PhD student Andrea Lopez Martinez, which I'm co-supervising with Susana. And her goal is really to put all that together, the new data tree, the data set, which is now complete, largely thanks to her. We have data for every one of these 1,200 species. And now she's conducting these very exciting analysis where the fossils are tips in the phylogeny. So stay put there. Hopefully there'll be more results in the future. All right, well, thank you so much for your attention. I hope that was clear, and I will be very happy to take any questions or comments.
Well, thank you very much for a great talk. We do have questions, so I'm going to start reading them. The first one is from Jorge Cortez, who's a professor at the Biology Institute. And it says, how do you integrate the interaction of angiosperms with their pollinators in the reconstruction of ancestral angiosperms and their diversification?
Yep, I'm just gonna write this down. I like, because I take note of the questions, that helps me think about the future. Unfortunately, we don't, and we can't, because there are insufficient data. Although there's an enormous amount of pollination data, these are not necessary for the same species that we have. We all know there's a lot of variation among different species of even the same genera, so we can't make those assumptions that what we know in one species applies to another. And also there's this difficult problem. I think you guys will know a lot more than I do, but it does not seem to be that common in the end that highly specialized relationships between plants and pollinators exist. There's a lot of generalized pollination systems, and that makes the questions even more difficult to answer, because angiosperms have not only shifted pollinators over and over, but they've also shifted from generalized towards more specialized systems, and occasionally back and forth. And so that there's no current really good comparative method that would allow us to do these inferences, even if we had the data. That's where we are now. The question is extremely important, and I hope that in the future we'll have enough data to start attempting solving that question.
Okay, I guess that would also answer another question that we had that said from Sofia Huerta, who is a student in our program, ecology program. So she said, with the reconstruction of the most recent ancestor of angiosperms, can you get an idea of the first pollinators? Well, at least that's related to the previous question. And then would it be useful to have information of the first floral visitors?
Yeah, absolutely. We're dying to have that information. Unfortunately, I think there are very, very few records of plant-pollinator interactions in the fossil record. There was a paper published, I think, last week or 2 weeks ago on florivory in a very early Cretaceous flower, and it was quite exciting. But these are like anecdotal, like it's just point observations. It's just one observation of probably many things that were already happening back then. So to answer the first question, do we know what the ancestral pollinator was? Absolutely not. We shall mention that among early diverging lineages of flowering plants, so the anagrade, Amborella nymphaealis astrobalearis, pollinators of living plants are quite well known, in fact, and these tend to include a lot of thrips, flies, and beetles. So these are like the three main groups of insects that have been talked about for basal angiosperms. I think bee pollination and butterfly pollination came about a little later in the history of flowering plants. But we can't derive what we see today in these lineages to their ancestors 100 million years ago, for various reasons. First of all, because shifts have occurred, keep occurring all the time. And second of all, because the fauna was different, has evolved since then. And so even if we had pollination data on every single one of living species of flowering plants, it would still be very hard to make that inference, because not the same groups were in the same proportions in those regions where the ancestral flowering plants were. That being said, I think the fact that we found it to be bisexual, and I didn't mention it, but also with a perianth, that's a very strong result, very likely to have had a perianth. That says a lot about the likelihood that it was in all likelihood, in my opinion, doing something with someone else. It doesn't make a lot of sense to be bisexual unless you're being pollinated by someone else. It's a high risk to be bisexual. There are strategies to avoid self-fertilization, but it's still a high risk. And it's also a high cost, probably, to produce a perianth. So these are two key characters that we interpret as having a role in the interaction with the pollinators, which probably were insects, but no one really knows. I think there's common understanding that it's more likely to have been insects than birds or mammals. But how many? Maybe it was a whole group of different orders coming in. Why they were coming, it's not entirely clear. They may just have come for a meal, like eat floral parts, eat pollen, not necessarily with that. There was probably no nectar. We don't know that for sure at all. But nectar glands have evolved many times in the flowering plants. There's no nectar glands in most of the early diverging flowering plants. But that just shows how many questions remain. So I would love to know, but we don't know yet. It was probably something or a group of somethings.
Okay, thanks. The next
maybe I should. Sorry, I should add something as well. Is that a lot of us have often assumed that the ancestor of the ancestral flower was wind pollinated, because most of the living seed plants are wind pollinated, except for cycads and some of the Nitalis. But there's very strong evidence now from the fossil record that many groups of extinct gymnosperms were already insect pollinated. So insect pollination is definitely not a key feature of flowering plants. But the way that flowering plants have done it might have been different and novel. So I just wanted to mention this before I forgot. Sorry.
All right, thank you. I think I'm going to read the question, one more question that's related to pollinators as well, and then I'll read the other questions. So this is from Miranda Ramirez, who was a former PhD student with us in the program of biological sciences. So she says, both symmetry and world fusion are associated with specialized pollination in extant species. Do you think that changes in floral development were paired with some pollinator radiations?
I would say yes, probably. I mean, what we know, so I haven't talked about symmetry at all, but I'm very interested in that as well in some of the work that we've done. So symmetry is a remarkable feature that has changed many times during the evolutionary history of flowering plants. There's been, a former student counted at least 130 different origins of zygomorphy, of bilateral floral symmetry in flowering plants. And some recent papers have shown that this could be way more, in fact. So it is very likely that development just creates new morphs, fuses, frees, makes things bilateral, then flips back. But the reason why these characters are fixed and maintained over time and possibly act on diversification is probably because of their impact on something that they're doing with the function of the flower. And that being, in many cases, linked with pollination. So I don't think the pollinator comes, well, obviously the pollinator doesn't arrive and then say, be zygomorphic to help me out. But there's probably a lot of selection that has occurred in zygomorphic variants to maintain and make them even more zygomorphic. If that makes any sense?
Mhm.
Yes, I think so. All right. So the next question is by Packley Ortega, who is also a grad student in program Biological Sciences. He says, interesting talk with interesting proposals. Do you have any idea about the size of the ancestral flower of the Angiosperms?
No, but it was small. In all likelihood. So we didn't reconstruct size. Size is a highly variable character. So I didn't mention, but we chose, in general, quite conserved characters because these are the only ones that we could actually score in all of the flowering plants. So we excluded group-specific characters, but also in choosing more conserved characters, we also chose less variable characters. Because if a character is too variable, say changes very quickly, is different between closely related species, that for the models that we use, that means a high rate of evolution. The likelihood functions that we apply to these questions will find that a high rate can explain the data better than low rates. And so when you have a very high rate, you cannot infer the past. If size changes every minute, it's impossible to guess what happened millions of years ago. So for that reason, although I have not attempted it, and I'm not aware of anyone who's attempted it, I believe that if we did apply some models of trait evolution to floral size, it'd be really hard to get an answer. But that being said, what we do know is that nearly every fossil flower found from the Cretaceous is really tiny. They're really like microscopic flowers. There are exceptions, I think, but most of them are really small. And so I think that would probably suggest that there has been some increase in size of flowers through time in some groups. Obviously, some groups have remained very small or become secondarily smaller. That makes sense? But I would love to know.
Yeah, I know there's more questions, but I wanna follow up on that, which is related to the sizes. As you said, you know, in evolutionary time, size increased, right? As we know today, you know, those original flowers were small, and they you know increased. The more derived flowers increased in size. Now the organ and the gynoecium and the androecium also increased in size. So you know, there are probably several hypotheses for this. Would you say that it is more related to the pollinators, you know, for attraction, or to other mechanisms such as pollen competition, for example, that might be more related to a selection process within plants, or other mechanisms that may have caused this increase in size in evolutionary time? What would you say about that?
Probably nothing, because I would be really going outside of my expertise here. I know there's been quite a bit of interesting work very recently on floral economics. So on the physiology of flowers and the costs associated with putting more or less investments in carbon, nutrients in the production of flowers, including the relative size and contributions of different organs. but potentially I think this would have the power of addressing that question. But
I know it's a,
I do believe so. Maybe I can go back to something that I'm more comfortable with. So we were very, very careful not to compare our ancestral flower with any living flowering plants. People asked us, journalists asked us many times, can you tell us an example of similar flowers? And no, there's not. Maybe there are, but there's always different characters. You can't do that. In this, you can't find that flower nowadays. But why would you? Why would you have something which we might actually consider, it was brilliant in the Cretaceous, but that flower probably wouldn't survive 2 days nowadays. It was probably highly imperfect. It was just the first by chance construct that has evolved numerous times in numerous ways, but mostly, it probably had too many parts and they were not fused enough, etc., etc. And so I think in these 140 or more millions of years, flowers have become more efficient at doing what they do. Mm-hmm. And it is possible that through that history, for many of their pollinators, increasing the size has given them access to more pollinators. Mm-hmm. I mean, if you think about bird pollination, which is quite important in the neotropics at least, or here in Australia, that doesn't really work on tiny flowers, right? Mm-hmm. So sure. It's possible that there was a pressure from the pollination spectrum to increase the size and then have access to more pollinators, more strategies of specializing or generalizing. I'm not entirely sure.
Mm-hmm. Yeah, I mean, things like the increase in size of the style in itself, that is also, I mean, Mulcahy proposed in the 70s, that was probably related to pollen competition. So it was more like a mechanism for selection of better pollen, more related to the reproduction rather than the pollination in itself. But anyways, I mean, these are thoughts, and I thought of asking it anyways. I know there's not a clear answer for these kinds of things, but it's kind of fun to think about it and discuss it.
Absolutely, it's fascinating.
Yeah, thank you.
All right, I think we have one last question from the chat, from Karen Hernandez, and she says, what do you think were the main selective forces on the flowering plants to evolve to the pentamerous and differentiated worlds?
Okay, so I think for the number 5, I would be really hard pressed to give a good answer to that. I think there's probably good answers in development, and I am not an expert on that. But there's something funny about development, and I think paradoxically it comes from the Fibonacci numbers that are linked with the spiral series. And so it's been proven for a very long time that when you have spiral arrangement of parts, the total number, I can't even say that correctly, but there's a tendency of successive series to go in Fibonacci numbers. And these are 3, 5, 8, 13, 21, etc. And so even though flowers may have been ancestrally whorled, we're not sure, there is a spatial constraint for parts to be arranged in particular numbers. And a lot of flowers, actually, some flowers that are whorled, they're also developing a spiral, where they have what is said a successive initiation. Again, I'm not a really expert on that, but what we're looking here is at the anthesis. What's the arrangement of anthesis? But what's happening through development might be sometimes different. But there are mathematical reasons for which 3, 5, 8, etc. are stable numbers. So it's possible that the number 5 has been particularly stable for the spatial arrangement of parts, either pre-anthesis or post-anthesis. Just putting that down for the first question. The second one, what was it? Can you remind me? Pentamere and?
Oh, hold on a second. Oh, I see there's more questions now. But yeah, pentamere and differentiated worlds.
Yeah, differentiate. I think the question of differentiation is a little easier to answer. What we often teach is that sepals have a stronger role in protection of the bud prior to anthesis. And the petals, the corolla, have a stronger role in attraction. So it's easy to imagine from an ancestral flower that might be undifferentiated, that has found one way or another to create sterile decorations, to progressively separate these and assign different functions. Instead of you doing everything, you do that, you do that. So I think that's quite easily explained. Why we have a different, and that differentiation has occurred many times, not just in pentapetalae, but as you know, many monocots have differentiated as well.
Mhm.
Okay. Thank you. Well, we do have more questions, so actually several more. So this is from someone that doesn't have its full name, DMS Roshan. What I think is interesting is not if this model is right or not, but that it could be the basis to hypothesis to be tested. I guess that was a comment. Mm-hmm.
Yeah. Thank you for that.
And then we have one from Yvonne Herrerias Diego. She's a professor at Universidad Michoacana. She says, interesting talk. With this proposal, who is proposed as the pollinator or floral visitor?
Well,
that was again, maybe that was, maybe these questions were done before you answered, so that's already answered. Guillermo Huerta Ramos, have you considered reconstructing pollen traits? Are pollen fossils too problematic to constrain the models?
There are not. There's actually a lot of fossil pollen. There's many more than fossil flowers. And pollen traits are limited, but there are some. We can score the number of apertures, their shape. The sculpture of the surface of the pollen is highly variable, so that would probably be difficult to reconstruct. But there's other things that we can do. The size is variable as well. The main problem is that, so we've added some of these, we started to add these pollen characters to our paleo flower analysis, but that's because sometimes we find pollen in the fossil flowers. So that helps us constrain the positions of the fossils better. How pollen itself would help us inform the ancestral traits of the flower, that's not really clear. We don't understand enough about the correlation between pollen traits and floral traits to say anything, nor to a large extent the correlation between pollen traits and pollination. That would be a very interesting one to pursue, but every time I looked, there was never a really good relationship. There's this, people have said, well, small pollen is more likely to be wind pollinated, small and smooth pollen, whereas big pollen, highly sculptured pollen tends to be more insect pollinated. But the reality is there's been really, really few recent tests of these questions. That would be an interesting one to pursue.
Yeah, Guillermo also said, I think pollen traits would also provide info to back up a hypothesis on the ancestral pollinator or pollination system. Yeah, okay. So let me see. Maria Fernanda Martinez says, thank you so much for the talk. It's very interesting. Do you have any hypothesis about what were the first rewards in ancestral angiosperms? Pollinator rewards.
Yeah, so I don't really know, but I would, like intuitively, I would say it would be food. Not just the pollen, but also potentially the perion. Because the way I look at it and why I'm careful as well is because that occurs in magnolids and some early diverging angiosperms. So we can't draw that conclusion, but we know that this does occur. And so there's all those flowers with very thick tepals that are being eaten out. There's a lot of that's often associated with trapping, so temporary trapping of the insects, and then they eat their way out. And that often goes also with using the flower as a place to mate or as a nursery. So some insects lay their eggs in these flowers. They also tend to be warmer. So thermogenesis occurs quite a bit. We have no idea if it did occur in the ancestral flower, but it's sort of like very comfy little habitats that some insects have used besides food. Whether that played any role in the ancestral flower, I can't tell you. If it did, it probably looked very different from our model. It's not all widespread like this. Yeah.
So probably no nectar or volatiles, you say?
I thought there was nectar, but that's just because of my very superficial understanding of the distribution of nectar glands across flowering plants. But I thought that there was. Then we really don't know, to be honest. Yeah.
Okay, great. We also have Ismael Santos. The question is, was bisexuality in ancestral flowers key to their evolutionary success and current dominance?
I would just say yes. Yes, of course. Yeah, I mean, one clue of this is that unisexuality occurs for many reasons, but it's quite common in wind-pollinated plants. So it's, it's secondary, but it really shows that there was probably a very strong association between bisexuality and animal pollination, which seems to have been a good idea for flowering plants.
And it was probably self-compatible, right? Do you think? Yeah, very likely.
Well,
who knows? Yeah, I mean, I think that's another issue that would be interesting to reconstruct is all the self-incompatibility, right?
Yes.
Across
that's a whole other.
Yeah.
A lot of people are thinking about it.
Yeah.
Obviously, it wasn't like perfectly self-compatible and self-fertilizing. Otherwise, you just wouldn't have survived or diversified much, right? So it was probably a combination of both. How it put a break on that, it's uncertain. There's a lot of, I always get confused between dichogamy and hercogamy, but basically, I think it might have been protogynous because that occurs so much, but maybe it was also incompletely incomplete. It was not avoiding entirely self-fertilization because that's also a really good idea. I don't know. I invite you guys to read on that note. There's an incredible paper that was published a couple months ago in Current Biology about the relationship between claystogamy and zygomorphy by Simon Joly and Dan Schoen, and they show that Darwin suggested that a while back, but they sort of demonstrated it with comparative methods that claystogamy, so completely closed flowers that coexist with open flowers, that occurs more in hyper-specialized systems. and that is the definition of zygomorphy. There's more, pleistogamy occurs more in zygomorphy as a sort of way to self-fertilize when the structure of the flower prevents that. So what was happening in the early angiosperms, who knows? But it was probably not entirely outcrossing and not entirely self-fertilizing. But I really don't know.
Yeah, very interesting, all these things. Let me see. Paulina Cruz is the next question. Thank you very much for the talk. My question is if the hypothesis of the Jurassic gap has an effect on the construction of the ancestral flower.
Oh, so thank you for that question. So if I rephrase the question is because we don't know the age of the angiosperms, how does this uncertainty affect our reconstructions? So when we conducted our analysis on either on both trees that had angiosperms constrained at 140 million years and others at 220, 250 million years, we found exactly the same results. And I was first shocked by how little impact that big question had on that other big question we were trying to answer. But then thinking about it, it actually kind of makes sense because these two trees, they're just, one is stretched compared to the other. The time is taken into account in both cases. It's the same data, it's the same phylogeny. And just the fact that there's been more time proportionally over the entire flowering plant phylogeny doesn't really affect what we can reconstruct. I assume it would have a major effect if we had outgroups that we could score, but unfortunately flowers hardly exist outside of flowering plants. So the fact that we, in older hypotheses for the origin of flowering plants, the time between their seed, their stem node and their crown node is much smaller, and that would have an impact if the stem node was important. But unfortunately, we don't have data. We could potentially score the sex in the presence of a perianth, but that's about it. There's no equivalent for stamens, for carpels, etc.
Good. Now we move on, I think, to the last question. And this is Martin Hesahim de Santiago, who's also one of our graduate students. Thank you so much for the talk. It's very interesting. What do you think about the evolution of correlations between floral traits and the evolution of angiosperms from early to present species?
The correlation between the traits on one hand and the angiosperms on the other, is that? Or
I guess he's thinking about trait correlations and how these different floral traits do not evolve independently, but evolve.
Yeah. So it's a difficult question because on the one hand, we're almost certain that traits are not evolving independently, especially in the flower, that there's a very strong correlations. But on the other, these correlations are very difficult to take into account with the current methods that we have. So that was one question from one reviewer of the paper. And our solution to that, if you look up that paper, you'll see there's a figure or a table that answers that question. So we don't have any comparative methods that allow us to analyze correlations among many discrete traits together in the phylogeny. We can do 2 or 3 traits together, but then we just go into crazy matrices with horrible parameter space. So, and that's very different from the continuous traits where there's a lot of multivariate statistics that allow us to do that. So for the discrete traits that we had, because we couldn't do that, what we did is we looked at all of the pairwise correlations. So we looked at all of the possible combinations of 2 traits within our 20 or so traits. So that was more than 200 possible pairwise correlations. And we looked using comparative methods, both likelihood and Bayesian, whether or not we could detect a signal in correlation. So these are all, for those who know that literature, it's all derived from the original Pagel 1994 paper, which was looking at correlations among two traits. And then we looked at the impact of taking these correlations into account on our results. And we found two things. One was that correlation was everywhere. We found that more than 50% of the pairs were correlated. So there was an enormous signal for floral integration, which a lot of people had proposed for a very long time, but it was sort of like a massive confirmation of that, which really suggests that a lot of traits are co-evolving. But the interesting second conclusion was that we were getting the same overall results. So say if you took like bisexual flowers, so say bisexual and zygomorphy, bisexual and fusion, bisexual and number of carpels, etc., we would find in that case probably 99% of the cases that while taking the correlation into account, we reconstruct a combination of states that are saying the ancestral flower was bisexual. And so that gave us a form of reassurance that were we able to take properly all of those correlations into account, it is possible that our results would still be robust. We don't have the final answers, but the pairwise correlations are giving us a hint that perhaps our results are robust to that.
Yeah.
Thanks for that question.
Yeah, that's an interesting one too. Well, I think I don't see any more questions. Okay, Jacob said there's no more questions. So I guess that would be it. Thank you very much.
No problems. Thank you so much for listening and for all those questions. This was a really good experience. And don't hesitate to contact me if you have any other things you want to discuss.
Yeah, thank you very much. It was a great talk.